Hey there, welcome to Scrubbed In. Today we're diving into the life cycle of the human immunodeficiency virus, or HIV. Whether you're prepping for your exams or just curious about how this virus works, stick around for a clear step-by-step breakdown of how HIV infects the body. Let's get started. So, what is HIV? HIV is a retrovirus that targets and weakens our immune system, specifically by attacking CD4 plus T cells, which are key players in defending the body against infections. Over time, if left untreated, HIV can progress to AIDS, or acquired immunodeficiency syndrome, leaving the body vulnerable to life-threatening infections. But how does HIV do this? Let's break it down. The first step is attachment and entry. HIV has surface proteins called GP120 and GP41 that latch onto the CD4 plus receptor found on the surface of the T cells. Once HIV locks onto the CD4 receptor, it interacts with another receptor called CCR5 or CXCR4. This interaction allows HIV to fuse with the T cell membrane and enter the cell. So once HIV gets inside the cell, things get really interesting and a bit tricky. Like I mentioned earlier, HIV is what we call a retrovirus. And the reason it's called this is because of how it copies its genetic material, depicted here as HIV RNA. Normally, in nature, genetic information flows from DNA to RNA and then to proteins. But retroviruses like HIV flip the script and go in reverse. Instead of going from DNA to RNA, they start with RNA and convert it back into DNA. That's where the name retrovirus comes from. This process is called reverse transcription. HIV brings its own special enzyme to do this, called reverse transcriptase. It's a key player in how HIV hijacks our cells. Now, here's the catch. Reverse transcriptase is not very accurate. When it's copying the viral RNA into DNA, it tends to make a lot of mistakes. Think of it as a typo in the genetic code. These mistakes are called point mutations, and they can change the viral proteins or cause other effects, sometimes helping the virus evade our immune system. So, after HIV enters the cell, it uses reverse transcriptase to convert RNA into DNA, which is a crucial step in the virus taking over the host cell's machinery and continue to replicate. Alright, now that HIV's RNA has been converted into DNA, the next stop is the nucleus of the T cell. Once inside, the viral DNA needs to become part of the host cell's DNA. This is where another key enzyme comes into play, called integrase. Integrase is like a molecular glue. It helps HIV's DNA insert, or as the name suggests, integrate itself into the T cell's genetic material, making HIV a permanent part of the cell. This is a crucial step because once integrated, every time the T cell replicates, it also copies the viral DNA, as it is now part of the genetic material of the cell, creating more viruses in the process. Now, here's where things take two different parts. I remember them as they both begin with the letter L: lytic replication or latency. In the lytic replication pathway, the virus quickly takes over the cell's machinery, forcing it to produce tons of new viral particles. These new viruses eventually burst out of the T cell, destroying it in the process, and then go on to infect other cells. But HIV can also enter a state of latency. In this phase, the viral DNA stays quietly integrated into the T cell's DNA without actively making new viruses. The virus can hide for long periods, avoiding detection by the immune system. This is why HIV can be so tricky to treat. It can remain dormant, only to reactivate later when conditions are right. So, after integration, HIV can either go full throttle and start making new viruses right away, or bide its time, staying hidden until it's ready to strike again. With the viral DNA integrated, the T cell now unknowingly begins producing viral RNA and proteins. This is the transcription and translation phase, where the host cell starts manufacturing components of new HIV particles. After enough viral proteins and RNA are made, they assemble at the surface of the T cell to form immature HIV particles. The final step in this process is called budding, where the new virus particles push out of the T cell membrane, ready to infect more cells. Finally, as the virus buds off, another enzyme called protease processes the viral proteins to make the new virus particle fully mature and infectious. This is the point where the virus is ready to invade more CD4 cells, repeating the process over and over, leading to the immune system damage. And that's the HIV life cycle in a nutshell. If you found this helpful, don't forget to give this video a thumbs up and subscribe for more medical content. Also, drop any questions you have in the comments. Thank you, and we'll see you in the next video.